My name is Claire Wells. Welcome to my presentation on the central nervous system. The image here is a representation of the connectome, which is basically a map of every long-distance axonal connection in the human brain. So these lines that you see in the image are representing bundles of axons running from one part of the brain to another. Just FYI. Right, so before we go on, I'll just briefly review the relationship between the central nervous system and the rest of the nervous system. The nervous system as a whole can be divided into the central and the peripheral nervous systems. The central nervous system is the brain and the spinal cord. The peripheral nervous system includes all the nerves that are running to and from the central nervous system. The peripheral nervous system is further divided into the sensory division and the motor division. For the sensory division, you have sensory information coming from sensory receptors and going into the central nervous system along nerves. And then the central nervous system will integrate that information, sort of interpret it, and decide what's to be done about it. And the central nervous system will then issue commands, motor commands, that travel out in the motor division of the peripheral nervous system, ultimately going to your muscles and or your glands so that you can respond to your environment. And before we get into the central nervous system proper, we need to talk about what surrounds it and how it's protected. Okay, so the brain and the spinal cord are actually the most protected organs in your entire body. That's because they're incredibly important. Most forms of homeostasis are regulated through the brain, and as well as your ability to move through your environment and interact with the environment. So the central nervous system is extremely important to keep protected. But in addition to that, it's also very fragile. As you may remember, nervous tissue does not contain protein fibers in its extracellular matrix, which is generally where the strength of extracellular matrix comes from. So since the extracellular matrix for nervous tissue is essentially nothing but ground substance, that makes the tissue extremely fragile and very easy to damage. So it requires a lot of protection. Accordingly, you have the central nervous system being encased entirely by bone. So of course the brain is encased in the skull, and then the spinal cord is running through your vertebral column, so that it's surrounded both by the bone of the vertebrae and by the tough cartilage in those intervertebral discs. So the nice thing about all that bony protection for the central nervous system is that it helps protect the central nervous system from impacts. But the drawback is that it makes the central nervous system vulnerable to swelling, because if that tissue starts to swell up, there's nowhere for it to go. It can't get larger because it's constrained by the bones that encase it. So if you have extensive swelling in the central nervous system, that's going to cause cells to die, basically. The brain will kind of be crushed against the skull, and some of the cells will die. So apart from the bones protecting the central nervous system, you have additional protection from the meninges, which are a layer of 3 membranes that surround the central nervous system. You also have the blood-brain barrier, which prevents substances from the blood from moving into the central nervous system. And then you have the ventricles, which are filled with CSF. Ventricles are open cavities inside the brain that also communicate with the central canal and the spinal cord, which is just a little tube, a hollow tube that runs the length of the spinal cord in the center of it. Those ventricles and the central canal together are filled up with cerebrospinal fluid, which is basically giving buoyancy and shock absorption to the central nervous system and helping remove waste. So I'll go through each of these elements in more detail. Starting with the meninges. So the meninges is again 3 membranes that surround the brain and the spinal cord. The outermost one, which is the thickest, is the dura mater. It's made of dense irregular connective tissue. It has 2 layers. the most superficial layer of the dura mater is actually considered part of the periosteum of the skull. Between, like, in certain places within the skull, those two layers separate, and then between them you have the dural sinuses, which is where cerebrospinal fluid drains. Beneath the dura mater, you have the subdural space, and then beneath that, you have the arachnoid mater, which is the middle membrane. It's also made of dense regular connective tissue, but it's much thinner. You have little pores going from the arachnoid mater, crossing the subdural space, and connecting with the dura mater and the dural sinuses specifically. So there's basically, yeah, just little holes connecting the arachnoid mater to those dural sinuses. Those are called arachnoid granulations, and that's how CSF drains into those dural sinuses. Then beneath the arachnoid mater, you have the subarachnoid space, which contains most of the blood vessels that would be supplying the brain with oxygen, and it's also filled with cerebrospinal fluid. Crossing the subarachnoid space, you have arachnoid trabeculae, which basically prop it up so that it doesn't collapse. And then beneath that, you have the pia mater. So that's both the thinnest membrane and the deepest membrane surrounding the brain. It's made of loose connective tissue. Wherever you have, I think I have an image of it on the next slide actually. Okay, I do. So right here on this side, you can see the dura mater on the top here, and then beneath it the thinner arachnoid mater, and then the subarachnoid space is right here. And you can see those trabeculae, the arachnoid trabeculae crossing it right there. And then you've got blood vessels running through that, and beneath it you have the pia mater, which is very thin. Wherever the blood vessels branch to send little arteries down into the brain tissue to supply it with oxygen, the pia mater actually follows those vessels. So those vessels are always contained technically within a space that's continuous with the subarachnoid space. And they're always separated from the nervous tissue itself by pia mater. So your blood vessels are never directly exposed to brain tissue. And that's part of the blood-brain barrier. Right, so on the other side here you can see the dura mater. This is the top layer of the dura mater, which is actually the periosteum for the skull. And then here's the bottom layer of the dura mater. And then here's the arachnoid mater. And finally beneath it, the pia mater, which is transparent and very thin. And then in the middle here, you can see a dural sinus. It's right here. So that would be where cerebrospinal fluid is draining. This whole subarachnoid space here would be filled with cerebrospinal fluid, which in certain spaces wherever you have a dural sinus, you're going to have those arachnoid granulations crossing the dura mater and draining that fluid into the dural sinus right here. Okay, now for the ventricles. So in the brain you have 4 ventricles that are all connected to each other, and then in the spinal cord you have the central canal. So let's see here. That would be a lateral ventricle. You have another lateral ventricle behind it on in the other hemisphere of the brain. Those are connected with this ventricle, the third ventricle, which is directly in the middle of the brain. The lateral ventricles actually curve around, so they continue around. And then the third ventricle at the bottom here connects with the fourth ventricle, which is actually just this space right here by the cerebellum. Right, so it's just this right here. And then that continues down into the central canal of the spinal cord. So the ventricles would be completely filled with cerebrospinal fluid. Right here at the base of the cerebellum, there's some little pathways that the cerebrospinal fluid can take to exit the ventricles and enter the subarachnoid space. The ventricles are going to be lined with ependymal cells, which is a special type of glial cell that's found in the central nervous system lining ventricles. And within these ventricles, you have specialized ependymal cells and found in the choroid plexus, where you have a lot of capillaries that are leaking plasma out into the ventricles, basically, and that's where the cerebrospinal fluid is formed at. So ultimately, it's derived from the blood in these parts of the ventricles called the choroid plexus, and then it circulates around the ventricles and exits into the subarachnoid space. It's kind of continually being made and then draining into the veins. So the whole volume of CSF that you have is replaced about twice a day. And here's an image of how that cerebrospinal fluid is made and is drained. So here you can see the choroid plexus in the third ventricle, and the arrows represent fluid exiting the choroid plexus coming into that ventricle. So that would be the cerebrospinal fluid being filtered out from the blood. And then it can travel around all the ventricles. It would exit down at the base of the cerebellum into the subarachnoid space, which surrounds the whole brain, but it also continues down around the spinal cord. And then wherever you have a dural sinus, so a space between those two layers of the dura mater, you're going to have your arachnoid granulations that allow the cerebrospinal fluid to enter the dural sinuses, and then those dural sinuses drain into the veins. So the CSF ultimately is coming from the blood and is returning to the blood as well. Okay, the blood-brain barrier. The blood-brain barrier is a barrier between the blood and the brain, as the name suggests. It's formed from three structural features. So first you have the specialized endothelium in capillaries that lines capillaries in the brain. So those capillaries are aligned with the simple squamous epithelium, so a single layer of flat cells that are connected to each other with a lot of tight junctions, which basically makes it impossible for fluid to leak between those cells, which means that if the fluid is going to exit the capillary and get into the brain, or if something within the fluid is going to exit the capillary and get into the brain, it has to be specifically transported across the endothelium by those cells that make it up. So nothing can sort of accidentally leak between the cells. The cells actually have to intentionally transport something across. So they have receptors for glucose that will allow them to pick it out of the bloodstream and transport it across their cell bodies, and then eject it into the brain, basically. So that's the tight junctions between endothelial cells lining the capillaries that serve the brain. That's the main feature that forms the blood-brain barrier. But you also have astrocytes, another type of glial cell in the brain. Wherever you have astrocytes near capillaries, they're going to send out their processes to the capillaries and kind of spread them along the surface of the capillary to form these things called astrocytic end feet, which you can see right here. So the capillaries are going to be kind of coated with those end feet of the astrocytes. So the astrocytes are, through their end feet, able to influence the endothelium surrounding those capillaries, which is one reason why the endothelial cells have so many tight junctions between them. The astrocytes have influenced them to make all those tight junctions. And then finally, you have the pia mater, which every larger vessel or vein in the blood that's not a capillary. It's going to be in a little space that's surrounded with the pia mater, so that that vessel is not actually directly exposed to nervous tissue ever. So those three factors are making up the blood-brain barrier, but it's mostly based on tight junctions between the endothelial cells that line the capillaries in the blood. The capillaries in the brain. So the main purpose of the blood-brain barrier is to keep stuff in your blood out of your brain. Of course, that can include toxins that you have in your blood sometimes, but also it includes just hormones that you have, because most neurotransmitters in the brain also function as hormones in the rest of your body. So if you have those hormones traveling in your blood, if they're able to actually enter the brain is going to treat them like neurotransmitters, and basically the signaling within the brain will get all kind of mixed up. The brain will think that messages are being sent, but actually it's just these hormones leaking over from the rest of the body. So that's one reason why it's really important to have that blood-brain barrier there. It's really good for, I mean, it's actually crucial for allowing the brain to function, but it does have some drawbacks, especially whenever we want to deliver drugs to the brain, because any drug that's going to be useful for a condition that affects the brain has to get into the brain. Therefore, it has to be specifically transported across the blood-brain barrier by those endothelial cells that line capillaries in the brain. So for instance, for Parkinson's, that is a disorder caused by dopamine neurons in the brain dying, and there's some movement circuits in your brain, which we're going to talk about later, that require those dopamine cells to be influencing like the way the circuit works in order for it to actually function. So if those dopamine neurons are dying, then your motor circuits are basically not going to work right, and you're not going to be able to do voluntary motions normally. So the most direct way to sort of treat Parkinson's would just be to give people dopamine. That is the neurotransmitter that they're lacking in their brain. So if you could just put it there in the brain, then their symptoms would stop. But dopamine is also a hormone in the periphery, so it is not, or in the rest of the body, so it is not transported across the blood-brain barrier. So you can't use it as a drug for Parkinson's. So instead what they do is they use L-DOPA, which is the precursor for dopamine. So L-DOPA, because it's a precursor, is specifically transported across the blood-brain barrier by the endothelial cells. So L-DOPA will be taken into the brain, and then whatever dopamine neurons the person has left will be able to use that L-DOPA to synthesize dopamine. So they'll basically make extra dopamine, kind of making up for the dopamine cells that had died. But of course the problem with that is that, once all of your dopamine neurons are dead, there aren't any neurons left that would be able to use L-DOPA to make dopamine. And at that point, of course, the treatment doesn't work. Okay, so all of the protective features that the brain has to keep it safe, the spinal cord has as well, and they're mostly just the same. So it's also surrounded by three meninges: the dura mater, the arachnoid mater, and the pia mater. In this image here, the dura mater is the outmost layer here. The arachnoid mater is just beneath it, and then you have the pia mater is right against the actual spinal cord itself, and it's really thin, so you can't see it very much, but you can see the little ligaments that connect it to the arachnoid mater. So this space between the pia mater and the arachnoid mater is the subarachnoid space, and that is filled with cerebrospinal fluid, as well as the central canal in the center of the spinal cord. Then wherever you have nerves exiting the spinal cord, the dura mater and the arachnoid mater follow those nerves, and they form some of the membranes that surround those nerves in the peripheral nervous system and keep them safe and protected. The pia mater itself, you can see it going out into the spinal nerves right there, but it doesn't surround the outside of those nerves. Usually, if we want to see what's in the spinal fluid, the cerebral spinal fluid, we're going to sample that from, well, I guess always we would sample that from the spinal cord through the intervertebral discs, so that you don't have to drill a hole through bone to get at it. Because the cerebral spinal fluid in the spinal cord is continuous with the cerebral spinal fluid in the brain, sampling it from the spinal cord can be really useful to diagnose things that are going wrong with the brain. Because any molecules that are associated with a condition, a neurological condition that you have, they're probably going to end up in the cerebral spinal fluid since it's removing waste from the brain. And since it's all the cerebral spinal fluid so all the cerebral spinal fluid is in communication, therefore any substance in one part of the cerebral spinal fluid will be in the rest of it, so you can sample it in the spinal cord and find out what substances are circulating in the brain. And again, in the spinal cord you have essentially the blood-brain barrier, but it has a slightly different name, the blood-spinal cord barrier, since it's around the spinal cord. And again, it's mostly composed of tight junctions between endothelial cells lining capillaries that serve the spinal cord. But you also see the astrocytic infeed. Okay, now we can go into the actual parts of the central nervous system. So the brain is divided into 4 big parts, and then each of those big parts has smaller parts within it, and each of those smaller parts has smaller parts, and then those parts generally have subparts as well. So there's lots of levels of structures in the brain, and we'll only talk about the first couple of levels. Yeah, but there's like hundreds of different structures in the brain. So the 4 main parts of the brain are the cerebrum, the diencephalon, the cerebellum, and the brainstem. The cerebrum is the big part here. That is functioning in all of your higher functions and basically like your personality. So that's the part that allows you to use logic and critical thinking. It allows you to interact with your fellow human beings, and it allows you to exert self-control, and it allows you to produce and understand language as well. The cerebrum also is what allows you to interpret stimuli. So as visual information, for instance, is coming in from your eyes, the cerebrum is what's going to actually take that raw data and turn it into an image that you're able to perceive. And not only that, but then every object in the image will be identified by the cerebrum. So the cerebrum is crucial for your being able to perceive the world around you and understand what you're perceiving. And it's also crucial for initiating voluntary motor actions. So the cerebrum is the part of the brain where you decide what you want to do, and then a motor command is generated that will actually make your muscles do that thing. And finally, the cerebrum handles emotions as well, which means basically that the cerebrum is accounting for most things that you think of as making up your personality. The cerebrum is the part that's most sort of uniquely you. Then you have the diencephalon, which is this kind of blue ball in the center of the cerebrum. It would be, of course, within the cerebrum, so it's deep to the cerebrum. The diencephalon does some information processing, but for the most part, most of it is relaying sensory information from the spinal cord to the cerebrum, and also relaying motor information from the cerebrum to the spinal cord. So for most of your senses, all of the information associated with them as it comes in from your sensory organs, it's going to stop in the diencephalon first. It'll be processed a little bit, and then it'll go on to the cerebrum, so it can be actually interpreted fully. Also, another part of the diencephalon integrates most of the functions in your brain. Well, at least very many of the functions in your brain together. And it basically kind of serves as a point of communication between your higher functions in the cerebrum and your lower functions in sort of the brainstem. Then you have the cerebellum, which is the red part kind of at the bottom of the brain, at the bottom and in the back. The cerebellum is all about motor coordination. So evolutionarily, that's extremely important. As you can imagine, if you're trying to hunt food or run away from a predator, you have to have a high degree of motor coordination to be successful. So the cerebellum has been very important in evolution. And then finally, you have the brainstem, which is this purple area here, which is coming out of the bottom of the diencephalon and then runs between that and the spinal cord. So the brainstem is handling all the most basic aspects of your homeostasis. So it is where you have control of your lungs and heart, and your reflexes and your autonomic motor system, which of course is the branch of the motor system, of the motor division of the peripheral nervous system, that controls your involuntary muscles and your glands. You also have some relay of information in the brainstem. Of course, sensory information coming up from the spinal cord has to pass through the brainstem on its way higher up, and motor information from the cerebrum has to pass through the brainstem on its way down. So the brainstem relays that information and also does some information processing on it. And the brainstem is also the source for most of what are called the classical neuromodulatory transmitters. That would include dopamine, serotonin, and epinephrine, or well, norepinephrine. Okay, so once again, this big part out here, that's the cerebrum, handling all of your higher functions, interpreting stimuli, initiating voluntary motor movements, and handling your emotions and your logic as well, so basically forming your personality. Then beneath it, you have the diencephalon relaying information to and from the cerebrum and the rest of the nervous system, and also integrating higher functions from the cerebrum with lower functions from the brainstem. Then you have your cerebellum right here, which is responsible for motor coordination for your voluntary motor movements. And then finally, the brainstem right here, also relaying information to and from the cerebrum and the rest of the nervous system below it, and also handling, well, controlling your heart, your lungs, and your autonomic nervous system. Okay, the cerebrum itself can be divided into the cortex and into subcortical structures. So the cortex is the top part of the cerebrum that has all the squiggles on it, and then beneath the cortex you have the subcortical structures. The cortex is divided into 4 lobes and the insula, and then there's a variety of subcortical structures that are beneath it, but the main ones are the basal nuclei, the amygdala, and the hippocampus. Then finally you have a number of white matter tracts connecting one side of the cerebrum with the other, so connecting the 2 hemispheres. The most prominent one is the corpus callosum. Okay, so getting into the different lobes of the cortex, you have the frontal lobe, which is about like so. That lobe is where you have your critical thinking and logic skills, as well as math skills. Also your self-control, your discipline, and that means also your politeness, since politeness is a form of self-control. You also have your voluntary motor control there in the frontal lobe. So in this light red region, that's your premotor cortex, so that's where you're going to be planning voluntary motor movements. and then the darker red strip that's just behind it right here, that is the primary motor cortex. That's where all your motor commands to your skeletal muscles are initiated from. And that also defines the end, the posterior end of the frontal lobe. Okay, next you have the parietal lobe that starts at this blue strip right here, and it continues back this way roughly to right there. Its posterior and inferior boundaries are a little they're a little ill-defined, or a little loosely defined. The parietal lobe is responsible for attention, so where you are directing your attention. Also, your sense of touch, your sense of balance, understanding language, and sort of interpreting sensory stimuli or perceiving stimuli. Interpreting and perceiving stimuli. So the blue strip in the front, that's your primary somatosensory cortex. So that's the part of the brain that you're using to interpret touch sensations. And then in this circle, you can't see it very well, but it's right here. This would be Wernicke's area, which is responsible for your ability to understand language. The rest of the parietal lobe is, for the most part, going to be involved with your attention, also spatial perception, so understanding how objects are related to each other in space, and then also just perceiving and interpreting sensory stimuli. So you have parts of the parietal lobe that are going to kind of identify, for instance, objects that are in an image that you're looking at, so within your field of vision. And then also kind of draw up your memories about those objects and your understanding of what they are. So in the parietal lobe, you would be identifying, for instance, a chair that is in your field of vision as being a chair. And then not only that, but you're going to, in the parietal lobe, access whatever memories you have about that chair. So you might recognize it as specifically your chair or your favorite chair. And that would be in the parietal lobe. In the temporal lobe, that's the one that's on the side. So that's right here. Kind of goes back roughly that much. Again, its posterior border is not well defined. The temporal lobe is responsible for your sense of hearing and smell and balance, in conjunction with the parietal lobe, and also for language comprehension in conjunction with the parietal lobe again, and for sensory perception and interpretation. So this little blue spot in the temporal lobe, that's your primary auditory cortex, so that's where you're interpreting, or that's where you're sort of assembling a soundscape or interpreting auditory information. And then once you've interpreted that information or perceived it, it would go out to other parts of the temporal lobe where you would identify what different sounds are and what's making them, and draw up any memories you have related to sources of sounds. Wernicke's area extends a little bit down into the temporal lobe, so both the temporal and the parietal lobes are responsible for language comprehension. Then you have the occipital lobe, that is at the back. It's roughly this. The occipital lobe is all about vision, so it contains your primary visual cortex, which is the little blue part there. That's where visual information is arriving at first from your eyes, and also is just kind of interpreting an image. So converting that raw data from your eyes into an actual image that you can perceive, and then the information would go out into the parietal and temporal lobes, so that objects in the image can be identified. There's also the insula, which you can't actually see in this image of the brain, but it's right kind of in this crack On the next slide, I have a picture of it. The insula is responsible for your sense of taste and your sense of your internal organs. So usually, any sensory information related to your internal organs, you don't really have conscious access to, but occasionally you do. So for instance, if you ate way too much, then you're perceiving a sense of fullness in your stomach, and you would be perceiving that in the insula itself. So this kind of roughly what those different lobes are associated with functionally, but there is overlap between the lobes. Right, so you don't have like for instance, Wernicke's area, the overlaps is mostly in the parietal lobe, but a little bit of it is in the temporal lobe. Right, so these lobes are just kind of things that we've defined, and actual circuitry within the brain is not necessarily organized with respect to the boundary lines of the lobes. Most of the cortex in the image consists of association areas, so everything that's kind of everything that's beige, and then most of the stuff that's a lighter red, most of that stuff is association areas. Association areas are parts of the cortex where information is converging from different sensory systems, or just different systems in general, and it's sort of all being interpreted together. So in the association areas is where you would identify objects in an image that you're looking at, so the chairs and the tables, but you're also going to be associating that with memories that you have about those objects. So the association areas are kind of crucial for the function of the cortex, and well, specifically the higher functions, such as interpretation, like sophisticated interpretation of stimuli, and logic, and stuff like that. Okay, so here's that image larger. I guess the insula is on the next slide. Right, so here's your frontal lobe stopping at the red strip, which is the primary motor cortex. So the frontal lobe, again, is involved with logic and planning and self-control and politeness or social graces. You also have speech production there, and control of your voluntary movements. So let's see, right here, right here. This is Broca's area, so that's the part of the frontal lobe that's involved with producing speech. So the muscle movements needed to actually produce speech. The kind of more shaded area here is your premotor cortex that's involved with planning voluntary motor actions. And then the red strip at the back is your primary motor cortex, where all of your voluntary motor commands are initiated from. Then just behind it, the parietal lobe starts and runs about roughly like that. That blue strip is your primary somatosensory cortex, so that's where all of your touch information is first arriving in the cortex, and then it will then be interpreted. The parietal lobe is also involved with attention, spatial awareness, so knowing where you are in space and where objects are in space, interpretation of stimuli and perception. Then you have the temporal lobe on the side here, about like so. That includes the little blue spot, which is your primary auditory cortex, which is where auditory information is first arriving to the cortex. So the temporal lobe would be involved with interpreting and perceiving that information. Also between the parietal lobe, or shared between the parietal lobe and the temporal lobe, you have your sense of balance, and also you have Wernicke's area responsible for comprehension of speech or language. Finally, in the back, roughly there, you have the occipital lobe, entirely devoted to visual processing, to processing information from your eyes. And then here, here you can see the insula. It's right here. So if you were to kind of pull down the temporal lobe, there's a little bit of cortex hiding inside of it, and that is the insula, where again you have your sense of taste and perception of your visceral organs. You may notice, well, of course, the most prominent aspect of the cortex would be all the squiggles that are on it. Those are gyri, and the little ridges, the little depressions between them are sulci. The purpose of those squiggles is, or the gyri and the sulci, is to increase the surface area of the cortex. Neurons in the cortex are arranged into columns, where every neuron in a column is related to the same function. So if you can increase the number of columns in the cortex, then you can increase the number of functions that the cortex can do, basically, or the processing power of the cortex. So we have a lot of those gyri and sulci, which gives us quite a large surface area for a cortex. The sheep brain has much fewer gyri and sulci, and they're not as pronounced, so they're not as deep. So its cortex has a lot less surface area. It doesn't need to do as much intense processing. Okay, then we have the subcortical parts of the cerebrum, which are the basal nuclei, the hippocampus, and the amygdala. If you're looking at an older source, you might see the basal nuclei referred to as the basal ganglia. The basal nuclei consists of the caudoputamen and the globus pallidus. The caudoputamen is also sometimes called the striatum. The function of the basal nuclei is to gate voluntary motor movement. So anytime you decide to do something, you're going to use your premotor cortex to plan that action, and then your primary motor cortex will generate the command signal that's going to go out to your muscles. But before it actually does, it's going to go to the basal nuclei first, and the basal nuclei has to let that signal through. Otherwise, you won't be able to do the actual movement. So the basal nuclei is supposed to allow you to start moving when you want to, and it's supposed to allow you to stop moving when you want to. The purpose of it is to reduce noise so that you're not moving on accident. The basal nuclei is the brain circuitry, is the brain circuit that that isn't functioning right in people who have Parkinson's disease. It needs to receive a dopamine input from the brainstem in order to function correctly. And in people who have Parkinson's, the neurons in their brainstem that make the dopamine are dying. So the basal nuclei doesn't get the dopamine that it needs to function right. And then as time goes on, people who have Parkinson's will not be able to start a movement when they want to. And once they get going, they won't be able to stop the movement when they want to. Then when all of their dopamine neurons are dead, then they would be basically paralyzed. And unable to start voluntary movements. Then you have the hippocampus and the amygdala, which are connected to each other. The hippocampus is, it functions in learning and memory. So it's important for your ability to learn and to form new memories. Memory is not stored in the hippocampus, it's stored in the cortex, but you have to have the hippocampus to learn and to form the new memories. Then the amygdala is primarily involved with processing emotions, so just allowing you to have and feel emotions. Both of them are connected together in what's called the limbic system, so some of you may have heard of the limbic system. It's a system in the brain that controls your emotions, and it's basically different structures that are all connected to each other that have some role in controlling emotions or in processing that's related to emotions. So the amygdala would be the heart of the limbic system. It's the primary place where emotions are processed in the brain. And then the hippocampus is closely connected to the amygdala. That way, your emotions can help you learn, basically. So if something happens that you have strong feelings about, you're going to remember that thing well, because of this connection between the hippocampus and the amygdala. Okay, and here's images of that. So let's see, right here, the green, the green Jeez. Okay, cool. These green structures are the basal nuclei. Right, so you have this light green thing, this whole thing is the striatum, or the caudoputamen, and then the dark green thing behind it is the globus pallidus, and together that's the basal nuclei that are involved with gating voluntary motor movement. So sort of deep to that, or inside, to the inside of that, you have the diencephalon, which is shown in blue right here. So here's your diencephalon, and then right on top of it, you would have your basal nuclei. This large orange structure is the hippocampus, and then the little pink ball at the end of it is the amygdala right there. So the hippocampus would basically be wrapping around the outside of the striatum right here. And again, the hippocampus is involved with learning and memory, and the amygdala is involved with emotions. Okay, now on to the diencephalon. There's 4 parts of the diencephalon, 4 large parts. Of course, each of those parts has its own subparts, and the subparts also have subparts. But you have 4 main structures, larger structures in the diencephalon. So you have the thalamus, the subthalamus, the epithalamus, and the hypothalamus. Of those, the thalamus and the hypothalamus are the most important. Your thalamus is your relay station, so all sensory information that's coming in from your body, up from the brainstem and going to the cortex, all of it stops in the thalamus first, except for smell. Smell goes directly into the cortex without stopping, but everything else, so touch, vision, taste, hearing, and balance, all of that is going to go through the thalamus before it gets to the cortex, which means the thalamus has the opportunity to sort of modify that sensory information as it passes through. Also, all voluntary motor information coming down from the cortex stops in the thalamus before it continues on down to the spinal cord and the nerves. So the thalamus has the opportunity to influence that motor information as well. So for motor information, that would start in the cortex, then it goes to the basal nuclei to be gated, and then it goes to the thalamus. And from there, it would proceed on down to the spinal cord. Then you have the subthalamus, which is beneath the thalamus. You have structures in the subthalamus that give input to the basal nuclei that sort of function with the basal nuclei, specifically the subthalamic nucleus. Then you have the epithalamus, which is on top of the thalamus. The epithalamus contains the pineal gland, which secretes melatonin, which is the hormone that allows you to sleep. The secretion of melatonin from the pineal gland is controlled by the hypothalamus, which is the last part of the diencephalon. The hypothalamus is also below the thalamus, but it's also below the subthalamus. And its role is basically to integrate your higher and your lower functions and to control hormones. So the hypothalamus is integrating almost any function you have that could influence homeostasis. It's controlling your temperature. Your body temperature is actually primarily controlled by the hypothalamus. It controls the secretion of hormones from the pituitary gland. It controls drinking. So when you're thirsty and when you drink, it helps control when you eat. Also helps control sleeping, and finally your autonomic nervous system. It helps control your autonomic nervous system. Oh, and reproductive behaviors. Reproductive behaviors are primarily controlled from the hypothalamus, so that's everything from mating to actually raising children. So in general, you can get by without your hypothalamus, but you can't get by quite right. You won't be quite right. So the brainstem is the primary source of control for your autonomic nervous system. And so, you know, that will still be functioning correctly. Will still be functioning if you don't have your hypothalamus. But your hypothalamus is what allows the function of the brainstem to be coordinated with the function of the cerebrum. So that's what allows your sort of complex understanding of your situation to influence how your body is being controlled by the autonomic nervous system. So it allows the functioning of the autonomic nervous system to be appropriate to whatever situation you're in, that you're understanding using your cerebrum. You also have the master clock of the brain located in the hypothalamus, which is the basis for all daily rhythms and all seasonal rhythms in the brain and in your behavior. So for certain species that have like a mating season, like a specific part of the year that they mate in, that's controlled by the master clock in the hypothalamus. It receives information from the eyes. Specifically, it receives blue light from the eyes to know when it's daylight and when it's night. So throughout the day, you would be receiving this blue light signal to the master clock in the hypothalamus, and that's how the clock knows that it's daytime. And then at night, when the blue light stops, that's when, you know, the master clock knows that it's nighttime now. And so whenever that blue light stops, that's when the hypothalamus tells the pineal gland to start manufacturing melatonin so you can go to sleep. So that worked well when we didn't have like sources of light at nighttime, but now we have a lot of light at night, and especially like screens have a lot of blue light that they emit. So looking at screens late at night can interrupt your production of melatonin because your master clock will basically think that it's still daytime. And that can interfere with your ability to sleep. So for your phones and all, they have apps that will filter out the blue light, so it's more like nighttime friendly. The hypothalamus is connected to the pituitary gland, so the pituitary gland is basically coming off the bottom of the hypothalamus, and the hypothalamus controls the release of hormones from the pituitary gland. So almost all hormones, it's not literally all, but almost all hormones are controlled ultimately by the hypothalamus through the pituitary gland. You also have parts of the hypothalamus that have a leaky blood-brain barrier, so that some substances from the blood can actually get into the brain in the hypothalamus. And that allows neurons in the hypothalamus to keep track of what nutrients are in your blood. So the hypothalamus has neurons that can sense the osmolarity of your blood and how much glucose it has, how much amino acids it has, how much fat it has. So the hypothalamus knows what your nutritional status is, and it uses that information to basically influence whether or not you are hungry. Right, so the hypothalamus is receiving information from all over, directly from the bloodstream. It's receiving information also from the brainstem and also from the cerebrum, and it's basically putting all of that information together to influence how different systems are controlling homeostasis, including your endocrine system and the autonomic nervous system. And it also has the master clock. Okay, so here's pictures of the diencephalon. So the thalamus is the biggest structure in the diencephalon. It's these big blue lobes. I guess here this is an anterior view, so this is looking at it from the front. This is looking down on it from above, and here's your thalamus again, which again is the relay station. All sensory information except for smell that's going into the cortex stops in the thalamus first, and all voluntary motor information that's coming out from the cortex and the basal nuclei stops in the thalamus before going to the spinal cord. Let's see, in your superior view, you can see the epithalamus right here. The pineal gland is sticking out the back of the epithalamus. Oops. And right, so of course you have melatonin released from the pineal gland to allow you to sleep. Then you have the subthalamus, and a side view of the diencephalon. You can see the subthalamus right here beneath the thalamus. And then sort of to the front of that, but also beneath the thalamus, you have the hypothalamus, which is involved with integrating higher and lower functions and controlling homeostasis. Right, so that controls the pituitary gland. The pituitary gland is right here. That's controlled by the hypothalamus. The hypothalamus has the master clock for your body, and it's also basically receiving information from your blood, from the brainstem, and from the cerebrum, and coordinating all of that information together to decide what should be done for homeostasis. And it controls, heavily influences your eating and your drinking, your reproductive behaviors, your sleep patterns, controls hormone release from the pituitary, and helps control the autonomic nervous system. Okay, then the cerebellum, in the back and at the base of the brain, it has its own cortex. It has the cerebellar cortex, which also is lined with ridges that again increase the surface area so that you can get more function in a shorter or in a smaller volume. It has several lobes, and beneath those lobes of the cerebellar cortex you have the cerebellar nuclei, which are balls of cell bodies that are involved with processing for the cerebellum. And right, so the cerebellum is receiving information about what your intended motor actions are. So as you have motor commands to your skeletal muscles that are coming ultimately from the cortex, that command signal is going to branch. Part of it is, part of it's going to go to your skeletal muscles, and then the other branch is going to go into the cerebellum. So the cerebellum knows what it is that you had meant to do. The cerebellum is also receiving sensory information, primarily proprioceptive information. So that would be information about how much tension is in your muscles and about the angle that your joints are at. So basically, where your body, like how your body is positioned, and how much strength or how much tension is in your muscles. And also on top of that, balance. The cerebellum is receiving sensory information about balance. So based on all that incoming information, the cerebellum can know what you meant to do and also what you actually did do. And the cerebellum is basically going to correct future motor signals to make sure that they are in line with what you wanted to do. So it's determining how far off you were from your intended action, and it'll correct future motor signals so that they will actually produce the intended action. So the cerebellum it handles motor coordination, but it also handles motor learning, and stores muscle memory, probably. So without the cerebellum, you wouldn't be able to learn to do any sophisticated motor task like riding, or playing like sports. And then finally, you have the brainstem. The brainstem is divided into three main parts, which of course have their subparts, and the subparts have subparts. The three main parts of the brainstem are the midbrain, the pons, and the medulla oblongata. Those are in order from superior to inferior. So the midbrain is basically at the bottom of the diencephalon. The pons is in the middle of the brainstem, and then the medulla oblongata is the sort of the most inferior part of the brainstem that actually merges with the spinal cord. The brainstem includes central pattern generators. A central pattern generator is a circuit of neurons that has intrinsic rhythmic activity. That means that the neurons are going to be firing in a rhythm without any outside input. So if you have that circuit completely disconnected from the rest of the brain, it'll make the same pattern of activity over and over again. So you have central pattern generators controlling certain stereotyped motor actions or actions that occur the same way every time you do them. So for instance, swallowing, the way that throat muscles have to coordinate in order for you to swallow is the same time every time you swallow. They have to contract and relax in the exact same pattern. So that's controlled by a central pattern generator in the brainstem. Right. So in the midbrain, you have the orienting responses, which is a sophisticated reflex. You have a visual orienting response and an auditory orienting response. For the visual orienting response, basically it's a reflex in which if you see something moving out of the corner of your eye, you will turn your head to look at it. So turning your head to look at something moving in the corner of your eye is an orienting response. You are responding to that movement by orienting to put your attention and your eyeballs on the stimulus. And then you have a similar thing for hearing. So if you hear a noise that's not in your field of vision, like sort of a loud or startling noise, you will turn your head to look at it. So the orienting responses are handled by the colliculi, the superior and inferior colliculi, which you can see at the top of the midbrain. You also have pain perception being kind of gated in the midbrain. So in the midbrain you have a structure called the para-aqueductal gray, which is responsible for letting pain signals go through to the cortex or not. Controlling pain and pain relief in the brain is, well, primarily occurs in the midbrain. You also have some structures in the midbrain that send information to the basal nuclei. Prominently, you have the substantia nigra, which is the primary source of dopamine in the brain. So most dopamine neurons in the brain are in the substantia nigra in the midbrain, and their axons go to the basal nuclei. So it's those substantia nigra neurons that make dopamine and send their axons to the basal nuclei that die progressively in people who have Parkinson's. Going down, you have the pons. The pons contains some circuitry that modifies the respiratory central pattern generator. That central pattern generator is located in the medulla oblongata. It controls your breathing. It allows you to breathe by coordinating the muscle movements that are necessary for breathing. And then in the pons, you have circuitry that's going to modify that to speed your breathing up or slow it down, depending on what you need. Right, so in the medulla oblongata, in addition to the respiratory central pattern generator, you have the cardiovascular control center, which controls basically your heart rate and sort of the functioning of your cardiovascular system. Well, so the heart rate and then the vasoconstriction in your blood vessels. You also have the central pattern generator for swallowing in the medulla oblongata. In ANP2, you guys will learn about the baroreceptor reflex. That's a reflex that controls your blood pressure. So in your aorta, you have some sensory receptors that sense your blood pressure, and then they send the information to the cardiovascular control center in the medulla oblongata, and then the information is used to modify your heart rate as appropriate. So if your blood pressure goes up, that's going to be sensed by the baroreceptors in the aorta, and then that information goes to the cardiovascular control center, and the cardiovascular control center will then signal your heart rate to slow down, which will then reduce your blood pressure back to a normal level. So those are sort of the functions that are localized in one part of the brainstem or another part, but you also have some functions that occur in all three parts. So sensory information that's coming in from the body and it's going up to the cerebrum has to pass through the brainstem first. And then also motor information that's coming down from the cerebrum going out to the body passes through the brainstem as well. Also, all of your information that's going into the cerebellum and coming out of the cerebellum, it's stopping off in the brainstem or being passed through the brainstem to get there. In the brainstem, you have your primary autonomic control. So the brainstem controls the activity of your autonomic nervous system, and the hypothalamus will influence how the brainstem is doing that, but the brainstem is the primary source of control for the ANS. You also have the cranial nerve nuclei. The cranial nerves are exiting, most of them exit from the brainstem. Those are nerves that go to kind of your face, your head, and your neck, with the exception of the vagus nerve, which goes down into the abdominal cavity as well. So each of those nerves, where it exits the brainstem, it has nuclei that are sending their axons out along the nerve, or else are processing information that's coming in from that nerve. And those nuclei are going to be located, in different parts of the brainstem. You also have the reticular formation in the brainstem. That's a group of many different nuclei in the brainstem. It actually has a lot of functions, but one of the main functions that it has is regulating your level of alertness and attention. So that is the reticular activating system within the reticular formation, and it's required to actually keep you awake and alert. So the parietal lobe is controlling where your attention is directed, and then the reticular formation in the brainstem is controlling how alert you are in general. So if you can turn that system off, the person will lose consciousness. So some anesthetics work by inhibiting the activity of the reticular activating system, and then the person will lose consciousness when it turns off. The reticular formation also includes the raphe nuclei. Those are the source of serotonin in the brain. So almost all serotonin neurons that you have are located in the raphe nuclei in the brainstem, and then their axons would travel out pretty much all over the whole rest of the brain. Right, so in general, all of your classical neuromodulatory transmitters, which again are serotonin, dopamine, norepinephrine, those are the main ones. There's also histamine. Those are all, well, with the exception of histamine, but serotonin, dopamine, and norepinephrine, those are all coming from the brainstem. So the neurons that make them are located in the brainstem, and then the axons for those neurons will travel out to the rest of the brain. So in general, the brainstem is concerned with some of your most basic functions, but it also can influence some of your more sophisticated functions using those neuromodulatory transmitters, the serotonin, dopamine, and norepinephrine. Okay, here's an image of the brainstem. So, of course, here's your diencephalon on the top, specifically your thalamus. Then going down from there, you have the brainstem. So first you have the midbrain here. Then inferior to that, you have the pons. The cerebellum would be connected to the pons there. And then inferior to that, you have the medulla oblongata, which then merges into the spinal cord. Right, so all of those are controlling. Well, they're passing information to and from the rest of the brain. They are controlling the autonomic nervous system. They have cranial nerve nuclei, so most of your cranial nerves exit from the brainstem, and each of them has at least one nucleus associated with it. You have your sources of serotonin, dopamine, and norepinephrine there in the brainstem. In the midbrain, you also have your orienting responses, so looking at something that is moving in the corner of your eyes, or looking at something that made a sound where you can see it. Also in the midbrain, you have pain perception, and the substantia nigra that gives input to the basal nuclei and is damaged in people with Parkinson's. Then in the medulla oblongata, you have your respiratory central pattern generator, your swallowing central pattern generator, and your cardiovascular control center. In the pons, you have circuitry that modifies the respiratory central pattern generator, so speeding up your breathing or slowing it down. And then finally, in the whole brainstem, you have the reticular formation, which is partly responsible for keeping you awake, or is needed to keep you awake. Okay, that's the brain. Now onto the spinal cord, which we won't talk about very much, but just a little bit. So the spinal cord, for the most part, is involved with relaying information from the body to the brain and from the brain to the body. Right, so mostly it just contains axons that are either coming in from the nerves of the body and going up to the brain, or else they're coming down from the brain and then they're going to go out in the nerves. But in the center of the spinal cord, you also have cell bodies forming like gray matter. And those cell bodies, some of them are involved with information processing. So some of them are interneurons, right, that are not actually connected to a muscle or to a gland or to a sensory receptor or to a sensory organ. Right, so in that gray matter of the spinal cord, you have some basic reflexes. So for instance, like if you touch a hot surface, there's going to be a pain signal generated that goes into the spinal cord, and at that point it's going to branch, and the signal will travel up to the brain, so you become aware of that pain on one branch, and then on the other branch it's going to connect to an interneuron, then to a motor neuron that's going out to the muscles of your arm, so that you can jerk your arm back and away from whatever you had touched. So that way, that's a reflex. So even as you're becoming aware of the fact that you touched something hot, you're already jerking away from it, so that so you can stop touching it faster. So it just helps you respond to the environment more quickly. You also have in the spinal cord gray matter central pattern generators for gait, so for walking and for running, and stuff like that, jogging. So they've actually done some pretty messed up experiments in cats where they cut the connections between the spinal cord and the brain of the cat. And if you take a cat like that and put it onto a treadmill, the cat will actually start walking on the treadmill. And then if you speed up the treadmill, the cat will speed up how it's walking or start running to keep up with the treadmill. So that's how they figured out that gait is actually controlled and coordinated from the spinal cord itself. So even without the brain, you already have, just in the spinal cord, enough processing ability to walk. The spinal cord is divided into 4 parts: the cervical spinal cord, the thoracic, the lumbar, and the sacral. The cervical part and the lumbar part are the thickest, because that's where you have all the neurons that are related to your arm nerves and your leg nerves. All along the spinal cord, you have nerves exiting that are part of the peripheral nervous system. So, right in the segment of the spinal cord that's shown here, you have your spinal nerves exiting. And then it also shows an interneuron right there in the spinal cord gray matter. So that would be involved with a reflex, or potentially with a central pattern generator for gait. Okay, those spinal nerves, they're used to divide the spinal cord into segments. Each segment of the spinal cord has 2 nerves exiting from it, one on each side. And then each spinal nerve has 2 roots. One root is anterior and one is posterior, where sometimes they're called ventral and dorsal. So one of those roots is carrying sensory axons and the other is carrying motor axons. So the dorsal root has sensory axons that are going to be entering the spinal cord, and then the ventral root has motor axons that are going to be exiting the spinal cord. And then those roots fuse together to make a nerve. Those nerves, again, are coated by the dura and arachnoid matter from the spinal cord itself, but not by the pia mater. And then one other thing that's good to know about the spinal cord is that it's actually shorter than the vertebral column. As you are developing, the vertebral column grows longer than the spinal cord does. So the sacral spinal cord segments are actually located more like in the thoracic region of your vertebral column. And then the nerves will end up continuing further down within the vertebral column to exit between the appropriate vertebrae. So, for instance, for your S1 nerves in the first sacral segment, that actual segment is located further up, maybe right here or so, in the spinal cord, with where you have your thoracic vertebrae. But then the nerves that are coming out of that segment are going to continue down in the vertebral column all the way down to the sacrum and exit at the beginning of the sacrum. So at the bottom of your vertebral column, you actually don't have the spinal cord in there. It's just nerves. And that spray of nerves coming out the bottom of the spinal cord is called the cauda equina, which means like the horse's tail. Within the spinal cord, you have gray matter in the center, as I mentioned, and then that's surrounded by white matter. The gray matter is divided into horns. So you have the anterior horns right here, one on each side, also sometimes called the ventral horns. You have motor neurons located in those horns. So these red dots here, those would be the cell bodies for motor neurons. And then those axons are going to exit along the anterior root or the ventral root of the spinal nerve and then continue in the actual spinal nerve proper to a muscle. In the posterior horns, which would be right here, those are also sometimes called the dorsal horns. There you have your sensory neurons. So I guess the actual sensory neuron would be located in the dorsal root ganglion, but then it's going to send its axon in to the spinal cord, and it will synapse in the posterior horns of the gray matter. On the cell body for a second order sensory neuron, that's basically just taking information. Technically, it is an interneuron, but it's called a second order sensory neuron because it's just taking information from an actual primary sensory neuron and then sending it up into the brain. So it's not actually doing information processing, it's just relaying the information. Then you also have a small lateral horn. It's right here, right here, right here, maybe a little bit of right here. The lateral horn contains motor neurons for the viscera. So the anterior horn is going to be motor neurons that are going to your skeletal muscles, and then in the lateral horn you would have the motor neurons that are going out to your visceral organs. So those motor neurons would be for the autonomic nervous system. You only see that in certain parts of the spinal cord, only in the thoracic and lumbar regions actually, which is where your sympathetic nervous system exits the spinal cord. For the white matter, it's divided into tracts that carry sort of organized information. So you'll have one tract that's carrying touch sensory information up the spinal cord into the brain. You would have another that's carrying motor information coming down from the brain going out to the body. You have another one for pain information. In general, the tracts are called funiculi. So you'll have the lateral funiculus right here that's carrying pain information going into the brain and also some voluntary motor information coming out. Okay, and here's our overview of all of the brain structures that we talked about. So you have the cerebrum is the biggest part of the brain, involved with higher functions, logic, emotion, personality, perception, and interpretation of stimuli, that sort of thing. And then so the diencephalon is a little hidden here, but it's this blue thing in the middle. There's your diencephalon. That would be involved with relaying information and integrating information. Then the brainstem, controlling the autonomic nervous system and your most basic functions. And then the cerebellum at the back, controlling motor coordination. So if we look at the subparts, for the cerebrum, you have the cortex, which is the outermost beige part shown here. That's divided into lobes. So you have the frontal lobe, the parietal lobe, the temporal lobe, and then the occipital lobe in the back. Frontal lobe handling logic primarily and voluntary motor actions. The parietal lobe handling attention, spatial awareness, and interpretation and perception, as well as touch sensation. The temporal lobe handling auditory information, and also interpreting stimuli and perception of stimuli. Parietal and temporal are sharing the sense of balance and language comprehension. The occipital lobe is just vision, visual perception, and then what's hidden is the insula. That would be located right in this little crack or fissure between the temporal lobe and the frontal and parietal lobes. In there you have the insula, handling the sense of taste and your sensation related to the visceral organs. Then you have the subcortical structures within the cerebrum. Right here is your basal nuclei, and then it wraps around. The basal nuclei gate voluntary motor movement, so they allow you to do things that you want to do and allow you to stop doing those things and just cut down on twitchiness. These blue structures on the outside of the basal ganglia are actually the ventricles. Those are the lateral ventricles, making and circulating CSF around the brain. Then the orange loop there, just inside the diencephalon, just inside the basal nuclei, that's the hippocampus, and the little pink ball at the end of it is the amygdala. Hippocampus is handling learning and memory, formation of memory, and then the amygdala handles emotions. Inside the basal nuclei, you have the diencephalon. In the very middle of the diencephalon, you have the lateral ventricle. Most of the diencephalon is the thalamus. That is a relay station. All sensory information except for smell that's going into the cerebrum stops in the thalamus. Stops off in the thalamus. All motor information coming down from the cerebrum stops off in the thalamus as well. Then you also have the hypothalamus beneath that. So the hypothalamus can be seen right here. That's connected to the pituitary gland, which isn't shown here, but it would be maybe right there. So the hypothalamus is controlling the secretion of hormones from the pituitary gland. It's integrating higher and lower functions for the maintenance of homeostasis. So it's influencing when you eat and drink, when you sleep, your reproductive behaviors, controls temperature regulation, helps control the autonomic nervous system, or modifies how the autonomic nervous system functions. Also contains the master clock, and is in control of the pituitary. Then your brainstem. The midbrain is this part right here. The midbrain contains the circuitry for orienting responses, turning your head to look at something that you saw out of the corner of your eye or heard somewhere that you're not looking at. It also can gate pain perception, and provides input to the basal nuclei. Then you have the pons right here that modifies the respiratory central pattern generator. And finally, the medulla oblongata at the bottom of the brainstem, merging into the spinal cord, that contains the respiratory central pattern generator and the swallowing CPG and the cardiovascular control center. All of the brainstem has nuclei for the cranial nerves and is controlling the autonomic nervous system, and has the reticular formation, which allows you to remain awake. And the brainstem is also the source for your transmitters, dopamine, serotonin, and norepinephrine, that heavily influence the way the rest of the brain functions. Then finally, you have the cerebellum right there, that is controlling motor coordination and allowing you to make muscle memory. And here's an image of the sheep brain cut in half. So many of these structures can be identified in the sheep brain as well. Here you have the cerebellum. Here's the medulla oblongata. Here's the pons. Here's your midbrain. The superior colliculus is right there. The inferior colliculus is right here. Those are the parts of the midbrain that handle the orienting responses. Here, coating the brain, you can see the dura mater. And here at the bottom of the brain, you can see the pituitary gland. So that pituitary gland is connected to the hypothalamus, which is roughly right there. At the front of the hypothalamus and at the bottom, you can see the optic chiasm right here. This is where the optic nerve is crossing the midline and then entering the brain. On top of the hypothalamus, you have the thalamus right there. And then you have, here's the cortex, of course. This white line here, that's the corpus callosum. And let's see, just in here, you have the lateral ventricle. And right here is your pineal gland that secretes melatonin. Alrighty, so that's the brain. Those are the parts of the brain. So thank you for listening to this presentation, and have a good day. And the final thing we'll talk about in this chapter are the cranial nerves, which emerge from the base of the brain. Although we're going to cover the cranial nerves in this chapter, we're actually not going to test over them until the next exam, because technically they belong to the peripheral nervous system as nerves, not part of the central nervous system, although they emerge from the central nervous system. So the cranial nerves all come out of the brain, and most of them have their roots in the brainstem. So that means that in the brainstem, for these nerves, you have kind of bundles or groups of cells, cell bodies, that send their axons out along the nerve. So that's kind of shown in this diagram here. There are, of course, 12 pairs of cranial nerves. Most of them have their roots in the spinal cord. You can see some of those nerves emerging from the brain here. Oh, sorry. Most of those nerves have their roots in the brainstem, and you can see them emerging here. And then in kind of reddish color, you can see the roots. So for instance, for this nerve, nerve 5, which is the trigeminal nerve, its root is right here. So right here you have a group of cells. Their cell bodies are all right here, and the axons for all those cells travel out of the brain in this particular cranial nerve. So the only cranial nerves that don't have their roots in the brainstem are nerves 1 and 2, which are the optic and olfactory nerves. And they have their roots kind of more forward in the brain. So the cranial nerves are kind of grouped based on what type of information they carry. So you have the sensory cranial nerves that are carrying just sensory information into the brain. Then you have the motor cranial nerves that are just carrying motor information out of the brain. Then you have the mixed nerves that are carrying both. So some of their information is going into the brain and some of it is coming out. So we're just going to go through each of the pairs of nerves in those groupings. So starting with these sensory cranial nerves. So first we have cranial nerve 1, which is the olfactory nerve. That is carrying your sense of smell into the brain. Then cranial nerve 2 is the optic nerve. That is carrying visual information from the eyes into the brain. Then you have cranial nerve 8, which is the vestibulocochlear nerve. That is carrying information from your ears into the brain. Your ears are sensing sounds, of course, but they actually also sense balance. So the vestibulo part of the vestibulocochlear nerve is carrying balance information into the brain, and the cochlear part is carrying hearing information or sound information into the brain. And you can see kind of diagrams of how those nerves travel in this table here. You don't really need to know all this detailed information in this table, but for the olfactory nerve, that is coming from the basically from the nose and traveling into the brain. Then you have the optic nerve is traveling from the eyes into the brain. And then here you have the vestibulocochlear nerve, which is traveling from the ear into the brain. And this part of the ear that's a big spiral, that's the part that's carrying, or that's like sensing sounds. And then this part up here that has the loops, that's the part that's sensing balance. So both of those forms of information are going into the brain on that nerve. Then we have the motor cranial nerves. The first one is cranial nerve 3, or the oculomotor nerve. That is controlling most of the muscles around your eye. You actually have 6 muscles around the eye that let you basically point it, exactly where you want to point it. So most of those, actually 4 of those 6, are controlled by the oculomotor nerve. And then the oculomotor nerve is also controlling your eyelids and the muscles inside the eye that control your pupil size and how the lens is shaped. The pupil size is controlling how much light gets into your eye, and then the lens shape is controlling the focus of the image. So it's going to change shape if you're looking at something up close versus if you're looking at something far away. So that's all controlled by the oculomotor nerve, nerve 3. Then you also have the trochlear nerve, which is cranial nerve 4, that controls another one of the eye muscles, and then the abducens nerve, nerve 6, also controls the last eye muscle, the last out of the 6 eye muscles. So those nerves are shown in this diagram here. Here you have the abducens nerve on the bottom controlling one particular eye muscle. The trochlear nerve is on the top controlling another particular eye muscle. The rest of these muscles are all controlled by the oculomotor nerve, which is in the middle. And you can see that nerve is also entering the eye to control the lens and the pupil, and it would also be controlling the eyelid as well. Then you also have a couple more motor cranial nerves, which are cranial nerve 11, which is the accessory nerve. It's also called the spinal accessory nerve. That nerve is kind of weird because like part of it comes out of the brainstem, but part of it is coming out of the spinal cord. So that's why they call it the spinal accessory nerve. It's still classed as a cranial nerve though, because part of it's coming out of the brainstem. It actually descends into the neck to control neck muscles and also some of the muscles in your shoulder and your upper back. So you do need to know the names of the cranial nerves. For the spinal accessory nerve, you can either remember it as spinal accessory nerve or just as the accessory nerve, whichever one works better for you. Just bear in mind, like there's a few different mnemonics out there to help you remember the names of the cranial nerves in order, and whether you're calling it the spinal accessory nerve or just the accessory nerve is going to affect the mnemonics that you use. Then the last motor cranial nerve is the hypoglossal nerve, which is cranial nerve 12, and that's controlling the tongue. So both of those nerves are shown here. Here you have the spinal accessory nerve, so you can see that a lot of it is actually coming off of the spinal cord, and then this bit is actually the only part that's coming out of the brainstem. Then that nerve is going to travel down into the neck and control, neck muscles, also some shoulder and back muscles. Then here you have cranial nerve 12, the hypoglossal, which is just traveling into the mouth to control the tongue. The rest of the cranial nerves are all mixed nerves, so they're carrying sensory information and they're carrying motor information. The first of those is the trigeminal nerve, which is cranial nerve 5. That's the biggest cranial nerve. It's called the trigeminal nerve because it has 3 major, large branches to it. It's controlling the jaw muscles, and it also carries somatosensation for the face, and somatosensation is just your sense of touch. So anytime you touch your face and you can feel it, that information is entering the brain on your trigeminal nerve. Then you have the facial nerve, which is cranial nerve 7. That is controlling all of the muscles in your face and the glands in your face, so like sweat glands, also like your tear glands, mucus and saliva glands, all those glands, and it also carries taste information from the tongue. So this would be the nerve that you're using to make facial expressions, and to salivate and to taste a lot of things. Actually, it doesn't carry all of your sense of taste. It is only carrying taste for the anterior 2/3 of the tongue, which would be, the front 2/3 of the tongue. So most of your sense of taste is going in on the facial nerve, but you also have other nerves that carry the rest of it. So the facial nerve isn't shown in this diagram. Both of these images are actually for the trigeminal nerve, but you can see the trigeminal nerve emerging from the brainstem here, and it has its 3 major branches that are going to different parts of the face. So it's all, this particular drawing is showing the branches of it that are carrying sensory information, touch information from the face into the brain, and then this is the part of it that's actually controlling your jaw muscles. But the facial nerve would also be traveling basically across the whole face like this because it's controlling all of your facial muscles except for the jaw muscles, of course. So actually the diagram for the facial nerve is here. This view of the facial nerve is showing the parts of it that are controlling glands. So here's a tear gland. It also goes into the nose to control mucus production there, and into your mouth to control saliva and mucus production. And then you can also see it innervating or going to the tongue to provide a sense of taste. And then here are the parts of the facial nerve that travel all across the face to control the facial muscles. The next mixed cranial nerve is the glossopharyngeal nerve, which is cranial nerve 9. That controls the muscles that allow you to swallow. It also controls some glands, including your largest salivary gland. It also carries some taste information and some visceral sensory information. So it's actually carrying taste information that comes from the posterior third of the tongue, so the back third of the tongue. All that sensory information goes in on the glossopharyngeal nerve. And also the visceral sensation is actually going to be coming from chemoreceptors basically in the neck. So it's actually, you're going to have a branch of that nerve that travels down into the neck. At a certain blood vessel in your neck called the carotid, you have the carotid body, which has chemoreceptors that are actually sensing the chemistry of your blood. They're primarily sensing the amount of oxygen that you have in your blood. And also right beside those chemoreceptors, you have what's called baroreceptors, which are pressure sensors basically, and they're monitoring your blood pressure in the carotid artery. So that information is all visceral sensory information, information about your blood pressure, information about the amount of oxygen in your blood, and that's all going into the brain on the glossopharyngeal nerve. Then the last mixed cranial nerve is the most important one out of all of the cranial nerves, and it's the vagus nerve or cranial nerve 10. So this nerve is controlling the muscles around your voice box, so that you can talk. It also controls a good part of your autonomic nervous system. So it carries a lot of visceral motor information, specifically for the parasympathetic branch of the autonomic nervous system. It's also carrying some taste information and some visceral sensory information. So this cranial nerve doesn't just travel down into the neck, it actually travels down, through the neck, through the chest, and into the abdomen. So it actually goes into the abdominal cavity. So it's very, very long and big, and it has a lot of branches. And it actually contacts most of the organs that you have in your thoracic cavity and your abdominal cavity. When a nerve is contacting an organ, we say that it innervates that organ. So it's supplying that organ with neural control, or with it's either controlling that organ or it's carrying sensory information from that organ. But in either case, it's innervating that organ by connecting it to the nervous system. The branches of the vagus nerve that travel to all of these different organs in your thoracic and abdominal cavities, those axons are primarily belonging to the parasympathetic division of the autonomic nervous system. So that means that it's carrying motor commands to all those organs, and those commands are related to resting and digesting. So it'll slow your heart rate down, slow your breathing rate down, increase the activity in your digestive system, stuff like that. So that basically is the reason why the vagus nerve is so important. The fact that it goes down into your thoracic and abdominal cavities and is innervating, carrying parasympathetic motor information to all those different organs in those cavities. That is why the vagus is such an important nerve. But it also does some other things as well. As I mentioned, it controls a voice box. It also carries taste information that comes from the back of your throat. You actually can taste a little bit with the back of your throat. That information all goes in on the vagus nerve. And it also carries some visceral sensory information that's coming from the aorta. So just like in the neck, you have the carotid body that is sensing the amount of oxygen in your blood and sensing your blood pressure. In the aorta, you have the aortic body that's also got chemoreceptors that are sensing the chemistry of your blood, and it has baroreceptors right there that are sensing your blood pressure. One difference between the chemoreceptors in the carotid body versus in the aortic body is the carotid body chemoreceptors are most sensitive to oxygen levels in the blood, whereas the aortic body chemoreceptors are most sensitive to carbon dioxide levels in the blood. Each of them can sense both, but they're more sensitive to one or the other. So here you have just a couple of mnemonics to help you remember the cranial nerves. There's also other ones that you can look up online. But one good one is, on old Olympus's towering top, a Finn and a German viewed some hops. So in this mnemonic, every first letter of each word is the first letter in the name of one of the cranial nerves, and they're in order from 1 to 12. And in this mnemonic, the S in some is for the spinal accessory nerve. So if you're remembering it as the accessory nerve, you might want to find a different mnemonic. Another mnemonic is, some say marry money, but my brother says big brains matter more. So in that one, the first letter of every word is standing for either sensory, motor, or both. So that's just to help you remember kind of the functions of the cranial nerves in order. Actually, for the test, you'll need to know the function more specifically than just sensory, motor, or like mixed. So you would need to know for instance, for the olfactory nerve, you need to know that it carries smell information, not just that it's a sensory nerve. So this mnemonic can kind of help you, but it's not, don't just remember the mnemonic and then don't learn like the other functions or the more detailed functions of the cranial nerves. It can just kind of help you retrieve the functions once you've learned them.